Hi folks, welcome to this Unit 1 revision video where we're going to look over some of the past paper questions that have featured from Unit 1, and we're going to go over the answers and the solutions and so on. This is a good video to watch just prior to doing your Unit 1 tests, either your Unit 1 unit test or the AB test that your teacher might give you as well. So if you're doing a Unit 1 whole Unit 1 test, or perhaps just before your prelims if you're focusing on Unit 1, or even your final exam, if you're only focusing on Unit 1, this is going to be good revision for you. So I've cut all of my Unit 1 questions together in this video, and I hope that you find it useful. Good luck. Hi there again. Welcome to the 2024 National 5 Biology paper run through. We're going to start off with paper 1 to begin with, and we're going to scroll down to question 1. So as always, 25 marks, 25 multiple choice questions, and the answers are A, B, C, or D for every question. So question 1. Cells in the stomach produce enzymes to help digest food. Identify the structure where these enzymes are produced. So this is a tricky first question, to be honest, in the paper, because there's a couple of things to consider here. First of all, you have to link two bits of knowledge. One is the 1.1 cell structure knowledge, which is the function of the ribosome, and you also have to link that to topic 1. Proteins, where you learn about the 5 different types of proteins, one of which is enzymes. So you basically have to replace the word enzymes for proteins in this question. And once you say identify the structure where these proteins are produced, it becomes much easier, and that then relies on your knowledge from 1.1, which is that the ribosome is the site of protein synthesis. So that would be the answer for the first one, is C. Question 2. The diagram shows cells viewed under a microscope at a magnification of times 100. Okay, so this is 100 times larger than it actually is. What is the average length in micrometers of the cells shown? So what we have to do first of all is we need to count the number of cells that are going across the field of view here, across the diameter. So that's going to be 1, 2, 3, 4, 5 cells there. Okay, 1, 2, 3, 4, 5. Then we're going to have to do a wee calculation, and we're going to have to get 1200, and we're going to divide that by the 5. That's the 5 cells going across. So 1200 divided by 5 is going to be 240. Okay. Now that gives us the width of, or the length of, one of the cells that are shown, 240 there, and they're asking us for the average length, so that would be fine. Now it says that it's magnified by a scale of times 100 here, but we can actually ignore that because the scale's already in. Okay, question 3. The diagrams represent the movement of molecules across a cell membrane. Which cell would require the greatest number of mitochondria to allow the movement shown? So the knowledge that we have to link in this question is between ATP production in 1.6 respiration, and obviously the function of the mitochondria, which is the site of aerobic respiration. And we also have to link that to active transport, which is our topic 1.2. So which cell would require the greatest number of mitochondria? Now, it's the cell which is going to need the most ATP, because ATP is produced in aerobic respiration. And the cell that would need the most ATP is the one that's carrying out the most active transport, because as we know from 1.2, active transport requires energy. So if we look at A, we've got a equal concentration both inside and outside the cell, so it's not that one. If we look at B, we've got a higher concentration outside than we do inside, and it moves inside, so that's simple passive transport. E, similarly, we've got a high concentration inside and a low outside, but again, it's passive transport because we're going from high to low. But if we look at D, we're going from a low concentration outside to a high concentration inside, so that would be active transport, and therefore your answer for this one is going to be D. Number 4, a section of DNA contains a total of 6,400 bases, of which 32% are guanine. The number of adenine bases in this section is. So what I always do for this question is I write down my 4 letters, A, T, C, and G, and I write down the numbers. Okay, so if there's a total of 6,400 and 32% are guanine, first of all, let's work out what 32% of 6,400 is. That's 2,000 6,448. Okay, and we can do that by multiplying 6,400 by 0.32, 32% as a decimal is 0.32. So 6,400 times 0.32 will give us 2,048. Now, because guanine is complementary to cytosine, that means we also have 2,048 cytosine as well. Okay, so what we do is we times that by 2 to get the total, which is 4,096 between cytosine and guanine. Okay, 4,096. And then we get the total, which is 6,400. We take away the 4,096 and we get 2,304. Now, that 2,304 has to be split between adenine and thymine, which are complementary to each other. So we divide the 2,304 by 2 and we get 1,152. Okay, and that's for both adenine and for thymine. Okay, so the answer is going to be A for this one, for question 4. Okay, so a little bit of maths involved in that one, but we managed to work our way through that. All right, so the key thing is, remember, complementary base pairing, A goes with T, C goes with G. Work out 32% of guanine. You know that cytosine is going to be complementary to that. Then you get that total, 4,096. When you total those two together and you take away from 6,400 and you're left with the 2,304, and then we half that again because we need 50% adenine and 50% thymine from the remainder there. Okay, number 5. Which diagram shows the concentration of product during a degradation reaction? Okay, so for this question here, we look at the graph and we have to work out if it's a degradation reaction. First of all, we are breaking down a substrate into a product. Okay, so a larger substrate into a smaller product. As time passes, a substrate is going to be broken down into more product, so we would expect the concentration of the product to continue to increase over time. Now, obviously, there are circumstances where, for example, you run out of substrate and therefore the product that's made is obviously going to slow down eventually. But this one is during the reaction, so it suggests that there's enough substrate available, which means it's going to be C for this one, where we see that continuous increase in product made over the course of the reaction. So C for that one. 6, the following stages occur during the process of genetic engineering. We have to put them into the correct order. So if we look at the 4 options, gene inserted into plasmid, plasmid cut open, plasmid extracted from bacterial cell, and gene extracted from chromosome. So we know that the first thing we have to do is we need to locate the gene on the source chromosome. That's not one of the options. Next thing we do is we extract that gene from the chromosome. So that's going to be number 4 there, isn't it? Which is going to come first. We then obviously have to get our bacterial cell and extract the plasmid, then cut the plasmid open. So that's going to be 3, then 2. And then finally, we insert our source gene into the plasmid and it becomes a modified plasmid. That's going to be 4, 3, 2, and 1. So the answer is going to be D there for number 6. You need to know your stages of genetic engineering to help you with that question. So memorize those if you are not sure yet. Number 7, a group of scientists investigated the effect of temperature on the growth of genetically modified cells. The GM cells were grown in a nutrient solution at 20 degrees. This was repeated at 30 and 40 degrees using fresh nutrient solutions at each temperature. Which would the following would improve the reliability of results? So if you watched any of my videos before, you'll know that I always teach the reliability as RIRI. So RIRI is reliability repeat. So we're looking for some sort of repetition here to increase the reliability within this one. So would we set up the nutrient solutions containing GM cells at 25 and 35 degrees? Well, all we're doing there is increasing the number of temperatures that we're using, not actually repeating the experiment more times at the temperatures we already had. So it's not going to be A. Number B is going to be set up 10 nutrient solutions using GM cells at each temperature. That sounds perfect to me. That sounds like the right answer. C, use the same concentration of nutrient solution at each temperature. That is a good validity step and measure, but it won't improve the reliability. And use the same types of GM cell at each temperature. Again, good for validity, but for reliability, we're looking at B there, setting up 10 nutrient solutions, okay? So that's what we're looking for number 7. Number 8, a respirometer was set up as shown to investigate respiration in maggots. Lovely. Okay, which of the following would be a suitable control for this investigation? So remember, with controls, we are looking for an apparatus that's set up in exactly the same way as the original experiment, but minus the factor that we're investigating, okay? And what we're investigating here is respiration in maggots. So it's likely that every other aspect of the experiment will have to be the same, except from the maggots themselves, okay? So we have a look at the 4 options there, and in D we've still got the maggots there, but we've lost the the chemical to absorb the carbon dioxide, so that's no good. For C, we've got glass beads to replace the maggots, which is probably quite good for a control. However, we've changed 2 things because we've also lost the chemical to absorb carbon dioxide, so that's no good. For B, we've got glass beads replacing the maggots, which is good. And we've got the chemical to absorb the carbon dioxide, which is also good. And everything else looks very similar to the first one, apart from the glass beads replacing the maggots. So we've changed just one variable there. That's ideal. And then for A, we can see they've switched the chemical and the maggots around, but that's just that's not actually going to work, that experiment there. We've not actually made that a control there, and what we've done is switch 2 things around in their places. That's all. So the answer is going to be B for this one. Okay, where we've replaced the factor we're investigating, i.e. the maggots, and them carrying out respiration, and we've replaced them with these glass beads here and kept everything else. Hi folks, welcome to the 2024 National 5 Biology paper. This is section 2 that we're going to be covering in this session. So let's rattle right on to question 1 and have a look at this. So section 2, as always, is worth 75 marks, and you have to attempt all questions. Hint number 1 from a biology teacher: do attempt all the questions. Don't leave things blank, even if you're not sure, because you never know if your guess is going to get you a mark or not. So question 1, Euglena is a single-celled organism found in water. The diagram shows some of the structures within the Euglena. We've got the chloroplast, we've got the cell membrane, and we've got another structure A. You have to identify structure A, which is the nucleus. Okay, next one, give the function of a chloroplast. So again, this should be a straightforward one for you if you know your cell organelles and their functions. That is the site of photosynthesis for the chloroplast. Have a wee look at 1.1 in your cell structures and definitions if you're not sure about those 2. It suggests why Euglena is not a typical plant cell. So there's a couple of reasons. First of all, it does not have a cell wall, which we find in a typical plant cell, and it has no vacuole. Okay, so the Euglena has no cell wall and no vacuole. They're both features which we tend to find in plant cells. Next question, the average body length of a Euglena is 50 micrometers. They can travel 1.3 body lengths in 1 second. Calculate how many micrometers a Euglena could travel in 30 seconds. Okay, so for the calculation here, we know that the body length of the Euglena is 50 micrometers. We know they travel 1.3 body lengths in 1 second, so we need to work out what 1.3 of 50 is. Okay, so we times 50 by 1.3 and we get 65. Okay, so that means that it travels 65 micrometers in 1 second. However, the question asks us how far can it travel in 30 seconds. So we're going to do the 65. We're going to then times that by 30. And we will get 1950. Okay, 1950 micrometers for that one. Okay, so hopefully you followed how we worked that one out. That's a wee bit of a problem-solving question there, isn't it? On to question 2. A group of students measured the mass of pieces of turnip tissue before placing them in different concentrations of salt solution. After 1 hour, the change in the mass of the turnip tissue was recorded. You have to predict the percentage change in mass in 2 grams per 100 centimeters cubed of salt solution. Now for this one, what we have to do is we need to look at the table and we can see that for a concentration of 1 and a concentration of 3, we know the percentage change in mass. So for a concentration of 2, we're going to go halfway between, because 2 is halfway between 1 and 3. For the percentage change, we're going to go halfway between 1 and 17, which is going to be 14. Because the change in mass can either be positive or negative, we would have to put a positive, a plus before the 14 there. So any value between 11 and 17 would be accepted by the SQA, but I'm going for 14 because it's right in the middle there. Part B, identify the salt solution where most of the turnip cells would be turgid. So if the turnip cells were turgid, that would mean that they'd gained a lot of water. And we know that plant cells become turgid when they gain a lot of water. So if you gain water, the mass of the cell is going to increase. And so we're looking for the concentration that causes the biggest increase in the mass of this turnip cell. And that, if we look at the table, is going to be plus 17, which is the concentration of 1 gram per 100 centimeters cubed. Okay, the pieces of turnip used in this investigation all had a starting mass of 6 grams. Calculate the final mass of the piece of turnip in the 8 gram per 100 centimeter cubed concentration of salt solution. So if we have a look at this one, we can see that the percentage change in mass is minus 9%. Okay, so what they're saying is at the start, it's 6 grams. When it goes into this concentration, it loses 9%. What will be the final mass? So we basically need to find out what 9% of 6 is, and then take that 9% away from the original 6 grams. So to work that out, there's a couple of ways to do it. You can go 6 divided by 100. That gives you 1%. Okay, so 6 divided by 100 gives you 0.06. That's 1%. We then multiply that by 9 to give us 9%, 5.4. And then we get our original value, which was 6, starting mass of 6 grams, and we take away 9%, which we've worked out as 0.54, and we should get 6 minus 0.54, we should get 5.46 for that one. So the correct answer, the final answer, is 5.46. Now, the SQA would allow you to round that up to 5.5. Okay, question 3. We're really motoring now. The diagram represents the processes that generate ATP in muscle cells. So hopefully we recognise aerobic respiration and fermentation there, which go in slightly different pathways depending on whether oxygen is present or absent. So let's have a look at the questions. Part 1, we have to name substance J and product K. So substance J is the product of the breakdown of glucose, okay, and the breakdown of glucose produces 2 molecules of pyruvate. Product K is the waste product produced at the end of aerobic respiration. Now we look at the other two, we've got carbon dioxide and ATP listed already. So the one that's missing there is going to be water, okay? So pyruvate and water. If we know our respiration reactions, then we should know those, okay? If you don't know them yet, I would suggest having a good look over 1.6 and memorizing those reactions so that you do know them. Okay, part 2, state the number of ATP molecules that would be produced from each molecule of glucose during fermentation. So we know that for fermentation, there is no second stage producing ATP. In aerobic respiration, the second stage produces a large number, but in fermentation, there is no ATP made. The only ATP made during fermentation happens during the first reaction, where glucose is broken down into pyruvate, and as well as 2 molecules of pyruvate, we also produce 2 molecules of ATP. Okay, again, that's just a memory one. You need to know that 2 ATP are made in that first breakdown reaction. Part B, name the cell structure where aerobic respiration is completed. So this is a throwback to 1.1 as well, to cell structure, and hopefully we remember that the mitochondria is the site of aerobic respiration. Okay, so that's what we're looking for there. Part C, it's a suggest question. These are the tougher ones. Suggest why a muscle cell might carry out fermentation rather than aerobic respiration. So what are the conditions for aerobic respiration? Well, we require oxygen to be present. So if a muscle cell carries out fermentation instead, that would suggest that there is no oxygen present, or oxygen is absent would be acceptable as well for that one. Okay, so well done if you did well on that question. If not, have a wee look over topic 1.6 again. Question 4, potatoes store sugar in the form of starch. Glucose 1-phosphate, or G1P for short, molecules are converted to starch in an enzyme-controlled reaction. This enzyme can be found in potato extract. Important, that sentence. Keep that in mind for later. So if we look at the diagram, we can see glucose 1-phosphate is being broken down by the enzyme into starch. In fact, I shouldn't have said broken down. It's actually been built up, isn't it? Because we've got the small glucose 1-phosphate molecules have been built up into one larger molecule of starch. And that's important because the first question asks us, identify the type of reaction carried out by this enzyme. This type of reaction is called a synthesis reaction. That's where we build up smaller molecules into one or several larger molecules. Identify the substrate in this reaction. Well, the substrate is what is acted upon by the enzyme, and the product is what is made at the end of the reaction. Okay, so in this case, it will be the glucose 1-phosphate, which is our substrate, and the starch, which is our product. Okay, so G1P or glucose 1-phosphate, you can write out the full thing if you like, is the answer there. Part B, an experiment was set up to investigate the time taken for starch to be produced. All previously made starch was removed from the potato extract. 2 rows of a dimple tile were set up as shown in the diagram. At 5-minute intervals, the content of the dimples at each column was tested for the presence of starch. The results are shown in the table. So we can see there's 2 rows. The first row has G1P, that's our substrate, and potato extract, which, if we read back to the top, is where we get our enzyme. So substrate plus enzyme equals an enzyme-controlled reaction. So therefore, we would expect to see a change in that first row there. In the second row, we have G1P in distilled water. Now, this time, we've removed the potato extract, which means we've removed the enzyme. And without an enzyme catalyzing that reaction, nothing is going to happen. Okay, and we can see that based on the results. The G1P in potato extract eventually detect a change and detect that starch is present. So the reaction's taking place because starch is our product. And the G1P plus distilled water shows no change, no starch is present. So therefore, no reaction has taken place. Now, that we've done all that analysis before we've even got to the questions, because it's important to understand what's happening in the experiment, because it will make answering the questions a bit easier. Okay, 4B part 1, suggest a reason for any previously made starch being removed from the potato extract. So we want to focus on starch being made during the experiment, okay? So that means that we've got to remove the starch so that only starch made during the experiment would be detected. We do not want starch that was made before the experiment contaminating our results or mixing up our results, okay? We have to prove that the starch was actually made during the experiment and not before. Part 2, state 2 variables that should be controlled to make this experiment valid. So let's have a look at the experiment. When I am teaching variables to my class, I always get them to remember the acronym MTVC, which stands for MTV channel. Now, when I was a youngster in the 80s, MTV was the best channel to watch. It was a music channel, and we all watched that. That when the number of channels we had were much more limited than now. So MTVC stands for mass, time and temperature, volume, and concentration. Now, if you can remember MTVC, you're going to remember the bulk of these variables we have to name in this question in the exam. Now, when we're dealing with solutions here, we're dealing with liquids, right? We're putting them onto a dimple tray, so it's liquids. And therefore, if we're thinking liquids, we're not probably thinking mass. They mention the time already in the experiment, so we're not thinking time here either. They don't actually mention the temperature here, and they don't mention the volume, and they don't mention the concentration, okay? Now, with the G1P, that is one solution. So we could say the volume of the G1P or the concentration of the G1P. We've also got potato extract, which is the enzyme. So again, we could say the volume of the potato extract or enzyme, or the concentration of the potato extract or enzyme. And lastly, we've also got distilled water in this experiment. So for distilled water, we could say the volume. We wouldn't necessarily say the concentration, because that should be the same, because if it's pure water, that concentration is just not going to vary. So the volume of, or the concentration of for this question, okay? And you could say any of the things, so the G1P, the extract, or the water for volume, and similarly for the concentration, we're saying G1P or the extract, or the SQA do allow you to say the enzyme instead of the extract for that one. Okay, so that is what they're looking for there. Those are the variables that would be controlled. Now, why is we mentioned temperature a moment ago. Why? Temperature is not mentioned in the experiment, but why is it not an acceptable answer here? Because simply controlling the temperature in the lab, in the room, is very difficult to do. You can't really control the temperature. You can measure it, but it's very difficult to control it. So therefore, that's why it's not allowed here. The volume and concentration we can control. Okay, part 3, 4B part 3, explain how the results show that the enzyme is required for the reaction to occur. Well, simply put, if we look at the results, when the enzyme is present, a reaction occurs, starch is produced. When the enzyme is not present, i.e. we've got distilled water instead of the extract, there is no reaction. Okay, so Starch is produced when either the extract, or you could say the enzyme, is present. Okay, that would be fine. Or you could say something like no starch is produced in the second row. Okay, and no starch is produced in the control experiment, because that's a control experiment, remember, where we've replaced the factor we're investigating, that's the enzyme, and we've replaced it with an alternative that will have no reaction, which is the water. Okay, moving on to question 5. The concentration of lactate in the blood of a long-distance cyclist was measured while they were cycling at different speeds. The graph shows these measurements at the start and the end of the competition season. Okay, perfecto. We've got the lactate concentration up the side here, and we've got the speed that they were cycling and the start and the end of the season. And we can see the start of the season has got a slightly greater upward curve, especially at those higher speeds and compared to the end of the season. So anyway, we'll see what the questions are asking us. Part A, what was the lactate concentration at the start of the season when the cyclist was travelling at 40 kilometres per hour? So start of the season is the solid line, and we go to 40 kilometres an hour, and we move up to the solid line, which is here, and we read all the way across the graph, and we can see that's sitting at around about 2.6. And we know it's 2.6 because that line's 2, that line there must be 3, because it's halfway between 2 and 4, and because there are 5 boxes between 2 and 3, that makes each box worth 0.2, and it's the third box up, so it's 2.2, 2.4, 2.6. Okay, so the answer is going to be 2.6 units there. Part B, calculate the percentage decrease in lactate concentration at the end of the season compared to the start of the season when the cyclist was travelling at 52 kilometres per hour. So it's the old percentage change question. We get one of those in every single exam. So I would say if you don't know this formula, remember it. Okay, it's going to help you so much when it comes to the exam. It's basically a free mark once you know how to do these well. So we're talking about the decrease between the end compared to the start when the cyclist was travelling at 52. So we go to 52 kilometres per hour. We go up to the end of the season, he was at 4. And at the start of the season, we were up at 6.4. So the change would be the 6.4 take away the 4. 6.4 take away 4, which is equal to 2.4. That's the change. We're going to divide that by the original. What was it at the start of the season here? It was at, what was it at? 6.4, wasn't it? Divided by the 6.4, then we're going to times that by 100, okay? So when we pop those numbers into a calculator, because even Mr. White cannot do this in his head, 2.4 divided by 6.4 times by 100 gives us 37.5, okay? So 37.5% is our answer there, okay? So the answer is really irrelevant in terms of us practicing here. The important thing is, do you know how to do the calculation regardless of the numbers or the situation you're given? Well, this formula here is going to help you get on the way and be able to do that. And as I say, there's always a question on percentage change, so it can be an increase or a decrease in every exam paper, so it's well worth knowing that. Okay, 5 part C. Calculate how many times greater the maximum lactate concentration was at the start of the season compared to the end of the season. So let's look at our maximums for the start and the end. We can see they're going to be here. So the maximum for the start was 9. Okay, 9. Go along here, 9. And for the end of the season, it was 5. Okay, if we go along here, that takes us to 5. So it goes from 9 to 5. Now, when you're asked how many times greater something is, we have to calculate how many times the smaller number goes into the bigger number. So it's a simple big divided by small, 9 divided by 5 in this occasion. Okay, 9 divided by 5 gives us 1.8. Okay, big divided by small when you're asked how many times greater something is than something else. All right. And remember, yes, we do our calculation, but we also put our answer on the box that they want us to put the answer in. Hi there, and welcome to the run through of the 2025 National 5 Biology past paper. We're going to start off with section 1, the multiple choice section, which is worth 25 marks. So there are 25 questions in total. Now, in this section of the paper, you will be choosing from 4 options, A, B, C, or D, and you'll be marking it onto a grid with a pencil. We'll run through it on the computer here, and we'll talk through not just the answers, but also the reasons for the answers and the working for some of the questions as well. Question 1, which of the following structures would not be found not be, you'll notice that in bold, found in a typical plant cell. So we've got the choice of chloroplast, cell membrane, plasmid, and mitochondrion. Now, hopefully you've learned that the chloroplast is part of a typical plant cell, so we can eliminate that one. The cell membrane is found in all cells, so therefore we can eliminate that one. The mitochondrion is found in most cells, including the plant cell, so we can eliminate that one. And C, the plasmid, is found only in the bacterial cell. So we know from our study of the bacterial cell that the plasmid is only found there and not in the typical plant cell. So the answer to the first question there is going to be C, plasmid. Question 2. Potato cylinders were placed into different concentrations of sucrose solution and any change in mass was recorded. The table shows the initial sucrose solutions inside potato cells and the concentrations of the solutions they were placed in. Which potato cylinder will gain the most mass? Now, aside from being one of the most exciting experiments you can do is, probably remember it from class where you bore up some potatoes and pop it in some water and leave it for a few days. Aside from that aspect, what this experiment does show is it shows osmosis taking place within plant cells. And so essentially what they're asking you here in this question is when it says which potato cylinder will gain the most mass, you're basically looking for the potato cylinder which gains water. Okay, now we know that osmosis is the movement of water molecules from a higher water concentration to a lower water concentration. So what we have to do here is we need to work out where the higher water concentrations would be and where the lower water concentrations would be. That's the first step. Now, if we look at the two columns, what it gives us is the sucrose concentration, not the water concentration. So what you have to do is you have to do a quick conversion back to water concentrations. So if we do a quick run through of all four options that we've got, A says that the sucrose solution inside the cell is 0.31 and the sucrose solution outside the cell is 0.45. So that tells us that we're going to have 99.69 inside the cell and 99.55 outside the cell in terms of water concentration. So 69 being bigger than 55 means that the water is going to move from inside the cell for A to outside of the cell, which means the cell is actually going to lose water. Therefore, it will not gain the most mass. Option B, we can see that the sucrose solutions are the same both inside and outside the cell. So therefore, there'll be no net movement of water molecules. For C, we can see that we get 0.31 again inside the cell and 0.25 outside the cell. Now this time we've got 99.69 inside the cell and 99.75 outside the cell. So we actually have a higher concentration outside of the cell this time, and that means that water is going to move from the higher outside to the lower concentration inside. So it is actually going to gain mass for C. But the question asks us which potato cylinder will gain the most mass. So if we look at D, we're going to have 0.35 inside, which is 99.65 water, and we're going to have 0.21 outside of the cell, and that's going to be 99.79. So 79 being bigger than 65 means that the water concentration is higher outside, and therefore water is going to move into the cell for D. And because the concentration gradient is greater for option D than it is for option C, more water will move into the potato and therefore it will gain the most mass. Okay, so we're looking for the greatest difference in concentration gradient there, and D has a slightly greater difference than C does. So that's why D would be the answer there. But that is a tricky question, and it involves a wee bit of working out there. Question 3. In a section of DNA, 34% of the bases are thymine. Which row in the table shows the percentages of the other bases in this section of DNA? So we know that, or hopefully we know, that thymine is complementary to adenine, and therefore we would expect the percentage of bases in this section of DNA to be the same for adenine as it is for thymine. And if we look at the table, options B and C both show that. So we know the answer has to be B or C. Now, if we combine the thymine and adenine together, that's 34 times 2. So that's going to be 68 for adenine and thymine in total. So therefore, that leaves us with 32 that are going to be shared between cytosine and guanine, because those two are complementary to each other. And with 32 divided by 2 is going to be 16. So there's going to be 16 cytosine and 16 guanine. So the answer we're going to look for here is C on this one. 34 adenine, complementary to thymine, which we're told is 34. That leaves over 32 to be shared between cytosine and guanine, so that's 16 each. Okay, so the answer for number 3 is going to be C. Question 4. Lipase is an enzyme that speeds up the breakdown of fats, producing an acid that decreases the pH. 3 test tubes were set up to investigate the action of lipase on fat in milk. So we get test tube R, which is milk and lipase, test tube S, which is milk and water, and test tube T, which is milk and boiled lipase. Note the boiled. The pH of each test tube was recorded at the start of the experiment and then again 20 minutes later. In which of the following test tube or tubes would the pH stay the same? So if the reaction is taking place, if lipase, the enzyme, is breaking down the fats, that means that the pH will become more acidic. That will decrease the pH. Okay, so if we look at our milk and lipase, now I would imagine that everything's going to function properly here, that the lipase is going to break down the fat in the milk, and therefore it will become more acidic. Okay, so therefore the pH should decrease in R. In S, we've got milk and water, so we're lacking the enzyme lipase this time. So I would imagine the pH would stay the same in this one. And then lastly, the milk and the boiled lipase. So we have the enzyme present this time, but because it's been boiled and therefore denatured, it will not be able to catalyze or speed up the breakdown of the fats, and therefore it will not become acidic, and therefore the pH should stay the same. So we're looking at S and T, which is option D for this particular question. Okay, R would become acidic because the lipase would break down the milk, but S and T would both stay the same. Question 5. A 100-gram sample of hemoglobin was found to contain 15 grams of leucine. What mass of leucine would be found in a 5-gram sample of hemoglobin? So a little bit of maths in this one. First thing we have to do is we have to find out how many times 5 grams will go into 100 grams. Okay, we need to scale this down. So if 100 grams of hemoglobin has 15 grams of leucine, how much would there be in 5 grams of hemoglobin? So 100 divided by 5 gives us 20. Okay, so that means that it should be 20 times less for the 5-gram sample of hemoglobin, okay, because it goes into 120 times. So what we then have to do is get the 15 grams of leucine, and we need to divide that by 20. And when you do 15 divided by 20, it should give you the answer of 0.75. Okay, so a little bit of scaling involved, a little bit of mass involved here, but that's how we would do it. 100 divided by 5 gives you 20. And then because we know it's 20 times smaller the sample, then we have to divide the 15 grams we found in the original sample by 20 to give us 0.75. Question 6. A culture of 350 bacterial cells was genetically engineered to produce a human protein. Only 210 cells successfully produced the protein. The percentage success was... So it's telling us there's a total of 350 bacterial cells, but only 210 were successful. So we have to work out 210 as a percentage of 350. So we do 210 divided by 350 times by 100, and that should give us an answer of 60%, which would be B there. Okay, so that's a simple percentage calculation. Question 7. Which of the following builds up in animal cells as a result of fermentation? So again, this is a straightforward knowledge question. There's nothing to work out here. We just either know this or we don't. Is it lactate, ethanol, glucose, or carbon dioxide? Now, we know that lactate is produced in animal cells as a result of fermentation. So immediately we suspect we know the answer there. But we can just eliminate the other ones. Ethanol is produced by plants and yeast during fermentation. Glucose is obviously the initial substrate, the initial thing that's broken down in the first stage of respiration. And carbon dioxide is a waste product in both aerobic respiration and fermentation in plants and yeast. So therefore is not produced by the animal cells. So the thing that you had to know here was how animal cell fermentation is different from plant cell and fungal cell fermentation. Question 8. The apparatus shown was set up to investigate the effect of temperature on fermentation in yeast. The investigation was carried out at 15 degrees and then repeated at 20 and 30 degrees. The number of gas bubbles produced was counted. Which two variables should be kept constant during the investigation? So the choices are temperature and time left for, the type of yeast and the time left for, the temperature and the carbon dioxide concentration, and the type of yeast and the carbon dioxide concentration. Now, it tells us in the description that the investigation was carried out at 3 different temperatures. So therefore, it doesn't make sense that temperature should be kept constant during the investigation. And because of that, we can rule out A and C. The type of yeast, that makes sense. We would want to keep the same type of yeast because different yeast will undergo fermentation at different rates. And the time left for, that makes sense because if we're counting the number of gas bubbles produced, if we have different times, then that's not going to be fair to compare those two. It will not be valid. And the type of yeast and the carbon dioxide concentration, well, the carbon dioxide concentration is not going to have any impact on the rate of fermentation. That one is irrelevant as well. So the answer is going to be B for this one. Okay, the type of yeast and the time left. Hi there, welcome to section 2 of the 2025 National 5 Biology paper. We're going to go through each of the questions in section 2 as well and just check the answers, but also explain where it's needed. So question 1A, students carried out an investigation to compare the ultrastructure of typical bacterial and fungal cells. Part 1, the table shows their results for the fungal cells. You have to complete the column for the bacterial cell by placing a tick in the appropriate boxes. So for the bacterial cell, we know that there are 3 cells that have got a cell wall: bacterial cells, fungal cells, and plant cells, so we can tick that one. We know that the bacterial cell is the only cell without a nucleus, so we're going to leave that blank. We know that bacterial cells don't have any membrane-bound organelles, i.e. any organelles that have a membrane, which a mitochondria does, so it does not have that. It has ribosomes because it does need to produce some proteins. It is the only cell which has a plasmid, and like all the other cells, it has a cell membrane. However, unlike the fungal and plant cells, it does not have a vacuole. Okay, so that's what we're looking for there. Those are fairly straightforward. They should be ones that you have memorized from your notes in terms of the structures of all the various cells and know which cells have which structures. Part 2, the structure of the cell wall in the fungal cell was found to be different to that of a plant cell. Name the structural carbohydrate that makes up a plant cell wall. We also learn this in the photosynthesis topic in 3.3, that the carbohydrate that's made for cell walls is cellulose. Okay, it also mentions it in topic 1.1, and it says simply that the bacterial and fungal cell are made of different materials, but the plant cell wall is made of cellulose. So that's another one where you needed to know that. That was a knowledge-based question there. 1B, on average, muscle cells contain 2,500 mitochondria, liver cells contain 2,000 mitochondria, and cheek cells can have 200. You have to calculate the simple whole number ratio of the number of mitochondria in these cells. Okay, so the first thing for this question that you should do is look at the way they want the answer displayed in terms of the ratio, and then write down your initial numbers in that order. Okay, so step 1, we'll get the numbers written down, the raw numbers that the question have given us. So that's 2,500, 2,000 and 200. Okay, and we just check they're in the right order as they should be in the answer, and that's fine. So then what we have to do is we have to find a common factor that they'll all divide into. There's one quick way of doing this, which is we look at the number of zeros at the end of them all, and they all have 2 zeros. So that means we can divide by 100 in all of them, and we'll very quickly get to this answer by just removing the 2 zeros at the end. Okay, so the answer would be 25, 20, and 2. Okay, so that's a little bit of a maths question, but hopefully that isn't too challenging for you. Explain why a muscle cell contains a large number of mitochondria. So muscles, we know, are involved in muscle contraction. So muscle contraction. Now, muscle contraction requires ATP, and ATP is our energy currency in the body. It's produced by our mitochondria. So there's a high energy demand. Okay, we get a high energy demand. So that would be enough to get you 1 mark for this question. It's worth 2 marks, you'll notice. And the second mark is for saying what I mentioned a moment ago, which is that the mitochondria is the site of ATP production. The mark scheme says you can get away with saying aerobic respiration. Okay, so something along these lines would be what you're needing for the 2 marks. Why does it have a large number of mitochondria? Because there's a high energy demand for that type of cell, for the muscle cell, because of muscle contraction, which we know requires ATP. And then we link it to a respiration topic where we know that mitochondria are the site of the second part of respiration, that aerobic part, and where we require oxygen. Okay, so that's your 2 marks. So let's have a look at question 2 here then. So the concentration of some ions within the cell sap of seaweed, the surrounding seawater, and a unicellular organism were measured. The results are shown in the bar chart. Okay, so we get three ions there, sodium, potassium chloride. The striped bar there is the cell sap of the seaweed. The central white bar is the seawater, and the right-hand shaded bar is the unicellular organism. Okay, fine. So part A, part 1, says name the ion that moves from the seaweed into the seawater by diffusion and give a reason for your choice. Straight away, we're thinking diffusion. We're moving from a higher concentration to a lower concentration. So let's establish what direction that is based on the question. So it moves from the seaweed into the seawater. So the seaweed is the diagonally striped bar, and the seawater is the white bar. So can you see any of the 3 ions where the diagonally striped bar is higher than the white bar? And immediately you can see that's going to be potassium as the ion. Okay, and what's the reason? Well, the reason is that there is a higher concentration of potassium in the seaweed and a lower concentration in the seawater. Okay, something along these lines. As long as you've given where the higher concentration or the lower concentration is, which indicates the direction of movement of the molecules, then you're going to get the mark for that one there. Next question, the unicellular organism must always have a sodium concentration of below 0.25 milligrams per litre. Name the process that moves excess sodium ions out of the unicellular organism. Okay, so let's look at sodium and the unicellular organism. Now, we can see from this shaded bar, it's at 0.15. Okay, so relatively low. The seawater around it has much more sodium in it. Okay, now by diffusion, sodium will be moving into this unicellular organism from higher to lower. So that's into the organism. So if it wants to move sodium ions out of itself, it will need to use the opposite method of transport, which is active transport. That's where we go from a lower concentration to a higher concentration. And lastly, the cell sap of the seaweed was tested to measure the concentration of each ion. Name the structure in seaweed cells that contains cell sap. So the cell sap is contained in the structure called the vacuole, and that's a fairly straightforward knowledge-based question for you to answer. Okay, question 3. The enzyme invertase is commonly used to make soft-centered chocolates. Sounds nice. Invertase speeds up the following reaction. Sucrose is catalyzed by invertase to produce glucose plus fructose. Part A, an investigation into the effect of pH on invertase at different temperatures was carried out, and the results are shown below. You can see them in the graph. Okay, so we have to identify the temperature at which invertase was the most active. Okay, so we've got 2 graphs here. We've got one showing 45 degrees, where the enzyme activity is significantly higher than at 55 degrees. So the temperature where it was most active is where there's the most enzyme activity, so therefore that's going to be 45 degrees. We've then to state the optimum pH of invertase, and again, if we look at both graphs, we can see the peak in enzyme activity is at pH 5 for both. So therefore we can happily say pH 5 is our answer there. 3A, part 3. Predict what would happen to the enzyme activity if the investigation was repeated at 75 degrees. Now, what it's asking you here is not what will happen to the enzyme, and a lot of students would quickly write down here the enzyme would be denatured, which is true, but the question is asking you to predict what would happen to the enzyme activity. So if the enzyme is denatured, the enzyme activity is going to either decrease or simply stop altogether. Okay, so there'll be less activity or no activity, less or no activity. Okay, so an answer along those lines would get you the mark for that one. The key thing is try and use the word activity when you're describing enzymes as often as possible. And when you're asked to predict what would happen, it's usually an increase or a decrease, depending on whether it's a temperature increase from a lower to a slightly higher temperature, or usually it's a decrease when you get in very high temperatures like 75 degrees. Part 4. Describe how this investigation could be changed to find a more exact optimum temperature of invertase. So this investigation was done at both 45 and 55 degrees. Now, the result of this was that 45 degrees obviously produces a much greater enzyme activity than 55 degrees. But what we don't know for sure is that if we had a temperature in between 45 and 55, would that in fact have been the optimum temperature? So what we'd want to do here is we would want to repeat, but this time with more temperatures, both above and below the optimum, which was 45, wasn't it? Okay, so we'd repeat with more temperatures both above and below the 45 degree. Okay, part B, a different enzyme speeds up the following reaction. Starch is broken down into maltose. Explain why invertase would not speed up this reaction. So the key here is to do with specificity of the enzyme to the substrate. We know that the active site has a particular shape on the enzyme, and that shape is specific to only one substrate. So in this case, invertase is specific, if we scroll all the way back up here, to sucrose. Okay, that's its substrate. So therefore, why would it not speed up this reaction? Because it is not specific to starch. Or you could say something like, it is only specific to sucrose. So like the inverted version of the answer there. Okay, and just if you wanted to improve that answer, you might say something like, rather than saying it is not specific, you would say, the active site is not specific to starch. That would be a better answer. And similarly, where I've written it here again, you could say the active site is only specific to sucrose. So something along those lines would get you the mark for that particular question. On to question 4. It says, interferon is a type of protein made naturally by cells of the immune system. It can be manufactured by genetic engineering and is used to treat viral infections. So interferon was one of the first human proteins to be manufactured by transferring genes between organisms. And that diagram there should be familiar to most of you from your topic on genetic engineering, and hopefully you recognise all the steps there. But the first question is a definition question. It's asking you what is a gene. And from your notes, you should know that a gene is a section of DNA that codes for a protein. Okay, section of DNA that codes for a protein. Okay, that's a simple memorization. You just have to learn the definition for the gene. Part 2, describe one of the uses of enzymes in the process of genetic engineering. So enzymes can be used to either extract or to, you could say, cut out the gene, or you could say it's used to insert the gene, okay? Or it's sometimes used to cut open the bacterial plasmid, okay? And you don't need to know the technical ins and outs of that, but basically by using the same enzyme, you're more likely to be able to stick the gene into the plasmid. It will have complementary bases there, but you don't need to know all that at National 5. Okay, so that's one of the uses of enzymes, anything from that list there. B, part 1. Before it's manufactured by genetic engineering, interferon was extracted from white blood cells. 35,000 litres of blood were needed to produce 400 grams of interferon. Calculate how many litres of blood were required to produce 1 gram of interferon. Okay, so what we're going to do here is we know how many litres of blood are needed for 400 grams of interferon, but we want to know how many would be needed for 1 gram of interferon. So we know the total grams of interferon for both here. So we simply say that 45,000 litres of blood is 400 times more than what we need for 1 gram of interferon. Okay, 400 grams of interferon needs 45,000. 1 gram, which is 400 times smaller, is going to need 400 times less than 45,000 litres of blood. So we do a calculation. We do 45,000 divided by 400, and that should give us 112. 400 should give us 112.5. Okay, and then we pop our answer in there, 112.5. Okay. So 400 grams of interferon for the first one. If we want to know 1 gram, that's 400 times less. So we then divide the 45,000 by 400. Okay, B, part 2. During its manufacture by genetic engineering, 85 milligrams of interferon can be produced per litre of bacterial culture. Calculate the mass in grams of interferon produced in 45,000 1,000 litres. Okay, so if in 1 litre, because it says per litre at the top there, we produce 85 milligrams, we have to calculate the mass in grams of interferon produced in 45,000. So the first thing we do is we say, right, we can produce 85 in 1 litre, and it's asking us for 45,000 litres. We times the 85 by 45,000. I'm going to have to use my calculator for that because my mental maths is not good enough to quickly do that. Okay, and that will give us quite a big number, 3.825 million. Okay, so a big number, but it also tells us that we want the answer in grams and that 1,000 milligrams is 1 gram here. So what we have to do is then we have to divide this number by 1,000. So when we divide that by 1,000, again, I'm just going to quickly do my calculator here. That would give us 3,825. Okay, and we go 3,825 grams as our final answer. Okay, so a couple of little stages in that calculation, but that's how we would work out something like that. Fortunately, they gave us the conversion there, so we knew we had to divide it by 1,000. Name another type of protein produced by the immune system in response to pathogens. Now, we know that our proteins are share, structural, hormonal, antibody, receptor, and enzymes. The type of proteins produced by the immune system, we should hopefully know from that topic, is the antibodies. Okay, that's again just a bit of knowledge there. And if you don't know your share for your proteins, then learn those as well. Not just what the acronym stands for, but also what the function of each of those proteins is. Okay, so question 5 is one of those questions where if you have done the hard work of memorizing your notes and your content and you feel confident going into the exam, this is the kind of question that should excite you because you're basically given an open book to write down everything that you've learned about that particular topic. So this one says, describe the breakdown of glucose in animal cells when oxygen is present. So the hint you're looking for there is basically oxygen is present, animal cells. So we're thinking aerobic respiration. Okay, so you're going to write down everything you know about aerobic respiration. So the first thing is that glucose is converted into pyruvate. Okay, 2 molecules of pyruvate. So that would get you 1 mark. Then the pyruvate is broken down into carbon dioxide, and you can use the chemical symbol here, that's fine, that's acceptable, and water. Okay. Finally, the 3rd mark for this first part is that we have energy being released. Okay. So that energy is released in the form of ATP. Okay. Energy released as ATP. Okay. And you could also add in here a large number. Okay, we don't need to give a precise number here. We just have to say a large number of ATP. If you wanted to say that in stage 1, as an alternative to this, you could say in stage 1, 2 ATP are produced, and you could actually say the value that's often quoted, which is 36 ATP being produced here as well, for a total of 38. But a large number is sufficient for your answer here. The other things that you could say are respiration begins in the cytoplasm, but for aerobic respiration, it's completed in the mitochondria. Okay, so it begins in the cytoplasm and is completed in the mitochondria. Okay, and I should say, and aerobic respiration, because although it does mention oxygen being present in the question, we should just make it clear to the marker that we understand that it's aerobic respiration. Okay, so that would also get you a mark. And the last thing to state is respiration is, and I get my classes to say this all the time, is an enzyme controlled series of reactions. Okay, so any mention of enzymes controlling this would also get you the mark there. Okay, so there's 5 points that you could put there. Any 4 of those 5 points would have got you the full marks in this question. Okay, so that's a nice one, I think.